Hello students, today I will talk about isolation of lymphocytes and macrophages. Introduction. Lymphocytes are small cells of about 7 to 8 micrometer in size. They constitute the white blood cells. They are produced in the bone marrow from hematopoietic stem cells and later transported in blood and lymph. Lymphocytes mainly compose of the T lymphocytes, also called T cells, and B lymphocytes, also called B cells. They play important role in immune response. They can be isolated from blood and cultured in vitro for various applications. It can be used for quantification of B cells and T cells to study histocompatibility antigens, etc. Macrophages are mononuclear phagocytic cells and plays significant role in both inert and adaptive immune response. Macrophages are derived from monocytes present in the blood. They are secretory cells which secretes varied cytokines and therefore involved in regulation of various immune responses. They are also involved in the pathology of autoimmune response. Isolation of lymphocytes from peripheral blood. Principle. Lymphocytes are isolated from blood by density gradient centrifugation. In this technique, bloods are collected in tubes containing anticoagulant. The blood samples are layered over fecal solution and centrifused for a short period of time. The cells in the blood samples will migrate differentially according to their density during centrifugation. Thus, different layers of cells will be formed. The lowermost layer will compose mainly of erythrocytes which have been aggregated by the fecal. Just above the erythrocyte layer will be the granulocytes which have migrated out of the fecal layer. Since the lymphocytes have low density, it will be present at the interface between the fecal layer and the plasma along with platelets and monocytes. Further, lymphocytes can be isolated from this interface layer with washing steps to remove any residual cells, fecal and plasma. Reagents. Diluent solution. Balanced salt solution can be used for dilution and washing purposes. Commercially available solutions like Hank's balanced salt solution, calcium magnesium free Dolbeck's phosphate buffer saline, or cell culture media, for example RPMI 1640, can also be used as diluents. Fecal pack media. Commercially available fecal pack or fecal histopack can be used as density gradient media. It is composed of fecal 400 sodium ditriazoids with calcium disodium EDTA. Fecal 400 is a synthetic high molecular weight polymer of sucrose and epichlorohydrin and readily dissolves in water. It is highly branched and spherical in shape. It has low intrinsic viscosity of about 17 ml per gram and low osmotic pressure. In the fecal pack media, it is present at the density of about 1.077 gram per ml. Sodium ditriazoid is the sodium salt of 3,5-diacetymido 2,4,6-diiodobenzoic acid. It is light sensitive and therefore the fecal pack media is stored in dark bottles. It facilitates formation of solution of low viscosity with high density in the fecal-packed medium. Steps in lymphocyte isolation. Step 1. Blood samples are collected freshly for better yield of the viable lymphocytes in 15 ml tubes containing EDTA. About 2 ml of the blood samples are diluted 1:1 with diluent solution and mixed by inverting the tube upside down. In step 2, fecal-packed media bottle is mixed by inverting the bottle several times prior to use. Then, about 4 ml of the fecal-packed media is pipetted out into a fresh centrifuge tube. In step 3, the diluted blood sample is then layered over the fecal-packed media slowly without disturbing the fecal layer. Care should be taken to ensure that the two solutions do not get mixed. In step 4, the tube containing the fecal media and blood sample is centrifuged in a swing bucket rotor at 400 G for about 30 minutes in a refrigerated centrifuge. The temperature of the centrifuge is maintained at about 18 to 20 degrees Celsius. In step 5, after the centrifugation, the tube is slowly removed from the centrifuge machine and the different layers of the cells are observed. Using a sterile pipette, the solution at the interface of the fecal layer and the plasma is slowly pipetted out into a fresh tube. This interface region is also called the buffy layer because of its appearance. In step 6, the volume of the isolated interface solution is estimated and about 3 times its volume of diluent solution is added for washing purpose. Then it is centrifuged at about 100 G for 15 minutes. This will remove the platelet present in the isolate. In step 7, the supernatant is then removed and the cell pellet suspended in about 6 to 8 ml of the diluent solution. It is then mixed by slowly pipetting the solution up and down. The tube is then centrifuged at 100 G for about 15 minutes. This second step of washing will remove most of the platelets in the isolate. Step 8. In this last step, the supernatant is removed and the cell pellet consisting of the lymphocytes are resuspended in an appropriate media for downstream applications. Isolation of macrophages from peritoneal cavity. Peritoneal cavity is present at the abdominal region of mammals. It is a membrane-bound, fluid-filled cavity within which the liver, spleen, gastrointestinal tracts, and other viscera are present. It contains many immune cells, including the naive macrophages, T cells, and B cells. Since it contains many macrophages, it is considered to be a major site for isolation of macrophages. Macrophages can be isolated from peritoneal cavity by two methods: direct isolation of peritoneal fluids containing macrophages and isolation of thioglycolate elicited macrophages. To isolate the macrophages directly from the peritoneal cavity of mouse, the animal is first sacrificed by cervical dislocation and mounted on a wax tray on its back. The abdominal region is then sprayed with 70% alcohol. Using a scissor and forceps, the outer skin is cut and the inner lining covering the peritoneal cavity is exposed. About 5 ml of ice cold PBS with 3% FCS is injected into the peritoneal cavity using a 27 G needle, and the peritoneum is gently massaged to release the macrophages attached to the peritoneal wall. Peritoneal fluid is then collected using a 25G needle attached to a 5 ml syringe by slowly moving the tip of the needle within the peritoneal cavity. This will prevent clogging of the needle with fat tissues and other organs. Maximum possible amount of peritoneal fluid is collected with the syringe needle. Later, an incision is made on the lining that covers the peritoneal cavity, and by using a Pasteur pipette, any remaining fluid is collected. During the procedure, if a visible blood contamination is observed, then the fluid is discarded. Then the collected peritoneal fluid is centrifuged at 1500 rpm for about 8 minutes. The supernatant is then discarded and the cell pellet is suspended in PVS or an appropriate media. For thioglycolate elicited macrophages, about 5 ml of 3% weight by volume brewer's thioglycolate medium is injected into the mouse prior to isolation of macrophages. After 3 to 5 days of thioglycolate injection, isolation of macrophages from the peritoneal cavity proceeded as described before. This method increases the yield of macrophages to up to about 10 times when compared to direct isolation of macrophages from peritoneal cavity. In this method, about 90% of the cells isolated from peritoneal cavity are macrophages. The main concern about this method is that the macrophages have some differences in their physiological properties. The isolated peritoneal cells containing the macrophages are then counted using a hemocytometer, and about 1.5 into 10⁶ cells are suspended in 1 ml of DMEM F1210 medium. The cells are then allowed to adhere by culturing for about 2 hours at 37 degrees Celsius in a carbon dioxide incubator. The adherent cells are 90% macrophages and the non-adherent cells are removed by simply washing with warm PVS. Isolation of peritoneal cavity cells yields macrophages. However, certain important points need to be noted. One important point is that the presence of blood contamination should be avoided to obtain pure population of macrophages. Euthanasia by cervical dislocation should be carefully done to avoid blood contamination in the peritoneal cavity fluid. During the collection of peritoneal cell suspension, it is important to avoid damage to bladder and any other organs in the peritoneal cavity. One advantage of this procedure is that a good number of naive unstimulated macrophages can be obtained for various applications. As a conclusion, lymphocytes and macrophages are important cells of the immune system. Isolated lymphocytes like T cells, B cells, and macrophages are widely used for studies in the immune system. Therefore, it is important to understand the methods of isolating these cells. The main method to isolate lymphocytes is density gradient centrifugation. In this method, fecal pack is used as a medium which separates cells in the blood according to their size and density. Macrophages can be separated directly from the peritoneal cavity or after thioglycolate elicitation from the peritoneal cavity. Thioglycolate elicitation increases the yield of macrophages, though these macrophages have some differences in the physiological properties. Thank you.